Hi, this is Ahmet Danli from Cold Spring Harbor Lab and Genome Research. This week we have the 86th Cold Spring Harbor Symposium on Genome Stability and Integrity. Joining me today is David MacAlpine from Duke University in North Carolina. Thanks for being here and welcome to Cold Spring Harbor. Before we start talking about your research, I'd like to briefly ask you about your background. Where you were born, what inspired you to become a scientist, and what got you interested in DNA replication?
Okay, fantastic. So, originally born in Omaha, Nebraska, but didn't live there long. My dad was in oil, so we lived all over the world, including Saudi Arabia, Libya, Peru, Holland. So it was quite the experience, but high school on was pretty much in Texas.
Yeah. And what got you excited about science?
So like many people, I went to undergraduate with the idea I was going to become a medical doctor, but then my grades were not really good at all. I had a little too much fun my freshman and sophomore years, and it wasn't until I discovered I had to, you were not forced to do, but as part of your degree, you had to do a rotation or, have practical lab experience. And once I was in the lab, it just all clicked, because now everything I'd learned in the biochemistry textbook, I'm now able to apply. But now I'm not at the base of the knowledge triangle, I'm at the apex. And just the joy of the discovery of getting some, a piece of film, I guess we don't do that anymore, but out of the autorat machine, and seeing the result and knowing something that nobody else knew, my boss didn't know, that just really excited me. And that drove me into graduate school. And of course, because my grades were so bad, I had to work my way up with a master's, and then actually work as a research technician, then a master's. Then I got into a doctoral program, and then I went to do my postdoc with Steve Bell at MIT.
And you ended up in DNA replication.
Right. So it was a long path towards replication, but it's always been the focus. So I worked on replication of rDNA and tetrahymena as a research assistant. Then I worked on mitochondrial DNA replication and segregation as a graduate student. Went to Steve's lab to learn hardcore biochemistry of DNA replication, but somehow I was the odd man out in the lab, and I started doing genomics with Drosophila, and that kind of set my career path.
It's never a straight path, huh?
It's never a straight path, but it's been a lot of fun.
So yesterday you talked about your well-defined system to study the uncoupling of helicase and polymerase at the replication origins.
Correct.
But for those who might have missed your talk, could you give us a summary, please?
Oh, of course. So we were interested in what happens when you activate the replicative helicase in the absence of DNA synthesis. And originally the question we were going after was, what happens to the local chromatin environment once a helicase begins unwinding DNA at the origin? We had shown several years ago that during G1, when the helicase is loaded, that there's a remodeling of the chromatin to accommodate the double hexamer of the helicase, but we really wanted to know what happened now as cells progressed into S phase with the initiation of DNA replication. Was there, with activation of the helicase, did you have eviction of specific nucleosomes? What was the pattern there? At least that was the project as I pitched it to the very talented graduate student, Rachel Hoffman, who did the work, and of course, she was able to take it in directions that I didn't expect. So she found because initiation is a very transient process, we had to have a way to block initiation. But so as soon as I initiate, DNA synthesis is going to begin. So she developed a very tight conditional allele of polymerase alpha, which is part of the priming complex. So you no longer get priming of DNA replication. And what she found is that the helicases, indeed, even with no DNA synthesis, just started unwinding DNA. And they went out for about a kb, which was really unexpected that they had that much oomph behind them without DNA replication behind the fork. And then we tried to figure out why but they eventually stalled. So that was the question, is why do the helicases stall? And work from John Diffley's group, Dirk Remus, have shown that checkpoints and Rad53 can be involved, and we try to look at this in an indirect way, but we found that blocking the intra-S phase checkpoint or not allowing Rad53 activation, the helicases still uncoupled and went out about a kb, and we considered a few other models, but ultimately I think for us it came down to GC content that the origin of replication itself is very AT-rich. So the helicase is easily able to unwind that. But it probably gets a little harder to unwind more GC-rich DNA, and that suggests that the helicase needs a functional replisome to have maximum processivity.
I was struck by the unreplicated gaps that you observed. How much variation do you see between different origins at different origins?
So this only occurs at early activating origins, but almost all the origins, the helicase goes out about a kb, and then once we let the helicase resume replicating DNA, the helicase is oriented to only go outward,
yep.
And there's nothing to synthesize the DNA in the middle, which left unreplicated gaps. But it was pretty uniform at all the origins of replication, and that caused catastrophic these cells just could not divide their DNA.
So can you speculate on how mechanistically that could lead to genome instability?
Right, so as the cells progress into, I should back up. When I said they couldn't, in the conditions we initially used, they experienced a delay in G2M, suggesting they had a problem resolving these unreplicated gaps. And we were able to identify additional helicases that were required to resolve them. But ultimately, the cells were unable to segregate and partition their genomes into daughter cells, and it just was a catastrophic mitotic event. And many of the cells, the chromosomes wouldn't separate, and they would just bounce back and forth between mother and daughter cell, and ultimately the cell would go ahead and divide with an anucleate cell, lacking a nucleus.
I thought your system using the micronuclease was very powerful.
Mm-hmm.
And I think you recently devised a method called Robocop, is that?
Yes.
Which actually includes more variables to model chromatin accessibility data.
Correct.
Could you tell us a little bit about that?
Yeah, so we've been using micrococcal nuclease and ATAC-seq to describe chromatin occupancy throughout the genome, but this is just a view of chromatin protein DNA occupancy. We actually don't know what proteins are bound, and that's where Robocop comes in. This was developed with a collaborator of mine, Alex Hardemink, who's a computer scientist, and it's a probabilistic model. It incorporates motifs, the accessibility, protein concentrations to actually predict what is being protected all across the genome. And we can run this in a dynamic way with time points to actually visualize how proteins are coming and going.
Is there a bottleneck in terms of what you can use the method on? Bigger genome? Or
right, so that's for our high resolution micrococcal nuclease chromatin occupancy, where we can actually see detailed chromatin maps at individual loci. The fundamental problem is the yeast genome is 200 times smaller than the mammalian genome. We have scaled it to the fly genome. It would scale to the mammalian genome. It would just take an inordinate amount of funding in sequencing to be able to get those high resolution maps like we have in yeast.
I thought that was a very smart method. I was curious whether you're also thinking of including perhaps transcription data, or maybe even throw in a couple of important histone marks.
Absolutely. So we can actually, just from our chromatin occupancy data, we can predict changes in gene expression. This was a paper we had in, Trung Tran was the senior author in Genome Research last year, that we can predict gene expression almost as well from just chromatin occupancy than the chromatin marks modifications themselves. So just by the nucleosome disorganization that occurs with highly transcribed genes, as well as the presence of transcription factors at promoters or their absence.
Are you thinking of including some other organisms for comparative analysis?
We would
maybe with not too big genomes.
Right. We're pretty much focused on Saccharomyces cerevisiae because we have all the genetic tools, but we do occasionally do some of these experiments in Drosophila, and that's only a 30-fold jump in genome size, and that's affordable at the moment, so we can do that.
I was curious about, now that you work on the chromatin structure around these origins, I was curious about potential transgenerational effects in areas of DNA replication that are not primary sequence related, but rather more epigenetic.
Absolutely, and that's where our laboratory is going right now. We've adapted this assay to look at chromatin assembly behind the replication fork, and we can combine that with histone chaperones, and we can actually see differences in deposition of parental and nascent histones behind the replication fork. And in some of the mutants, we can see the assembly time is different, and we think that's contributing to some of these epigenetic inheritance and cryptic transcription.
So that brings me to a rather naive question, I guess. To an organism, what are the advantages, disadvantages of having well-defined origins versus them being stochastic, as far as we can tell?
Right, so I think the field's understanding right now is development in higher eukaryotes requires reprogramming of the transcriptional environment depending on the developmental stage of the cell and where it is in development. And because the replication program is tightly coupled with transcription, it gives you a plastic way to remodel both of them.
That makes sense.
Yeah.
It's a very brief and nice answer.
Exactly. I can go on more, but people like David Gilbert would probably be better for that question, but it is definitely, I think it's tied that the transcription programs can be so different from cell type to cell type that if you define the origin in a specific manner, it would probably interrupt with that.
What about variations that you see in the population at the DNA replication proteins? What are the most prominent diseases or disorders that you can think of?
Right. So for the longest time, the DNA replication, at least with the hardcore proteins involved in setting up origins, they were all essential, and there were no mutants or disease associated with them other than, well, if you're not replicating, you're dead.
Yep.
But recently, a disease called Meyer-Gorlin syndrome, where the patient, it's a form of dwarfism, where that still not clear how replication impacts it, but almost all of the components that specify an origin have causal mutations for this disease.
I guess now that we are in the third day of the meeting, I can ask what you thought was particularly interesting or exciting, in this meeting and also in the DNA replication field in general.
Right. So for myself, the meeting's been absolutely fantastic. It's been the biochemistry and the structural biology, how it's completely transforming our, everything used to be a genetic view, but now we're really getting down to a molecular and atomic view of how these processes are happening. And that's been absolutely fantastic, seeing the work from, John Diffley and others. But one of the things that really also struck me is Stephen Eichler's talk on the completion of the long read sequencing, and now that we can do the structural variation and the segmental duplications, things that were completely blind to us in the genome builds is now accessible. And that was just the enthusiasm and passion he had for that project really just got me thinking to appreciate it all even more.
Well, you answered my next question, because I was going to ask you about the long reads and how those structural new data would actually impact what you're thinking.
And they're definitely impacting how we think about DNA replication, our ability to map origins and things along those lines. So it's really giving us a single molecule view of, or a single chromosome or replicon view of DNA replication. So it's been an extremely powerful transformation as well.
So what's going to be next for your lab, say in the next few years?
We're still really interested in chromatin occupancy, but we'd like to get to the point where we can do single cell analysis of this or single molecule. So we're thinking of ways to use long read sequencing, perhaps with methylation of specific bases that we can then read off, so with targeted methylation, that would then reveal protein occupancy. And that's something we're really looking to get into, so we could actually have a single molecule view, so we could see what the actually get a binary readout of chromatin occupancy. This factor is bound in what fraction of the cells exactly.
Yeah, that's going to be powerful.
Yeah.
Well, It was a pleasure talking to you.
It was a pleasure talking to you too.
Thank you very much,
Yeah.
and we'll see you at the next meeting.
All right, thank you. I look forward to it.
Thanks.
Bye.